Hi, happy Thursday. To this point, you have done your journal warm-up, you have worked on your apps, you've read an article about cell communication, and you took a quiz on cell communication. And so now you have hopefully about 25 to 30 minutes left. And at this point, we are finishing up with section 2.1. In fact, tomorrow is going to be the last day before your quiz. So today, we want to take a little while to practice more about the structures in a eukaryotic cell. A few days ago, you looked at a cheek cell. So everybody will be on page A16. On page A16, this gives you directions for how to go back and look at cheek cells again for a second time. It says to create figure 1, which you see here, draw one cheek cell magnified to take up the entire space. So you're going to be drawing a cheek cell, not that's this big, which is about how big it appears to your eye, but to take up the entire space. So that's step 1. So you could even check that off after you do it. And it says label the 3 structures of the cell that can be identified through the microscope. So when you're looking at the cheek cell through the microscope, then you will only see 3 structures. Now a reminder, you need to chew on your cheek a little bit, and then use the toothpick. This is to make the cheek cell slide. Use the toothpick, this groups them off. You put a drop of methylene blue on the slide, and then you put a cover slip on top and focus the microscope. And then you throw, of course, the toothpick away. Remember that when you are done, you do need to rinse the slide off and put it back on the cart, please. So you've been looking at a cheek cell. You labeled the 3 structures of the cell that can be identified under the microscope. But that doesn't really represent a eukaryotic cell. So next, you're going to use a different color to add other structures. And so these other structures, remember, we're talking about an animal cell, and an animal cell has 11 parts. So you should have 11 parts once you are done with figure 1. However, notice it does say a different color to add the other parts. We're going to change number 4 a little bit. It says using the pieces of colored paper, we're just going to use another color. Instead of having to worry about cutting and gluing, we're just going to use another color. So use a different color. And now you're going to identify where specific macromolecules would be found. Remember, your macromolecules are your P, C, L, and N. Proteins, carbs, lipids, and nucleic acids. So if I've already drawn the cell membrane, I know where some lipids are because I know that the cell membrane is made of phospholipids. So using a different color, I would say phospholipids. Now remember, you're drawing a cell that's much bigger so that you have a lot more space to draw in. Cheek cells are epithelial cells. They're found in areas of high wear and tear. They divide often. They have very limited differentiation. That's part of what you were talking about today, was the idea of differentiation. What you draw in figure 1 is just the model of any eukaryotic cell. So in figure 2, you're going to be choosing a different cell in the human body. You might choose things like a muscle cell. We've talked about those. You might choose a white blood cell. We've talked about those. You can choose any cell that you would like, but you have to know how it's differentiated. And in your notes, we talked about how muscle cells are differentiated and how white blood cells are differentiated. And you need to represent that with the quantity or number of organelles. So you're looking at a cheek slide under the microscope, and you're filling in what you think would be the organelles that that cheek cell would have. And then in figure 2, you're drawing your own representation of any differentiated cell. In figure 3, it actually wants you to draw the protective cells of the Epomia batis. This is actually the sweet potato. And what we mean by the protective cells is we actually mean the skin. And so what you're really drawing in figure 3 is you're drawing a plant cell. And then you'll tell me how a plant cell is different and similar to the human cheek cell. We're not going to look at the potato cell under the microscope today, but tomorrow we are doing a lab with the potato. And so it's important to start thinking about what exactly is a potato and how are its cells similar to my cells and different from my cells. So using the rest of the time that you have in class, you can set up the microscope and look at your cheek cells.